topic is written on the board. It is polymerase chain reaction. This topic is a common practice in molecular biology labs. Every day, almost in everyday practice, this PCR chain reaction is carried out in the labs. What is the reason of studying this reaction? See, in our studies, sometimes or many of the times, we happen to get DNA molecules or DNA segment or fragment rather is a very short one, and we need to study it. Now, to study that DNA which is in very less amount, it's a problem. Like we need to have something that can make a Xerox copy of it. Like when you are getting a document in your hand, sometimes or many of the times we need to have a duplicate copy, or I need to distribute the copies among many people. So what do? I make Xerox copies. So this PCR chain reaction is almost similar to making of the Xerox copies, that is polymerase chain reaction. It is helping us in amplification of genes. Amplification means making multiple copies of one DNA segment. Multiple copies you can make. The reason of making these multiple copies is that we need to have something which will give us so many copies that now our studies can be made much easier. Now, you may ask that what are the steps? Before I go on to the steps are basically very simple. It's not at all a headache to understand the steps. There are some prerequisites, there are some requirements to study PCR. But those requirements, as I say, that those requirements I will be covering in another video tutorial. Let me first brief you the steps. What are the steps that are required in PCR chain reaction? Polymerase chain reaction is actually making polymers, making many nucleotides. That in DNA, we know the basic structure of DNA is a double helical structure. Now this double helical structure, what happens is, first step 1 will be denaturation, that is making it separate. How to make it separate? That is the process of making separate two DNA strands to be separated is denaturation, and this is done in every day in the molecular biology labs. Now there are some requirements to do and some cautions also to be maintained. Let us study in this video in brief. In my next video, this will be going through a detail how the primers are acting, what are the primers, how this attachment will take place. That I will cover in my next video. So in this video, my prime focus will be on the steps. The first step already I have written, it is denaturation or the melting of the DNA. Double helical structure, it has to melt and made separate so that our next step could be made easier. So first step is denaturation. So let us see what is the next requirement for this process. We know that in PCR reaction, the prime activity is the second step, that will be primers will be added. Now there are enzymes. Names of some enzymes are there. What are those enzymes? Those enzymes are DNA polymerase and RNA polymerase. These enzymes are helping us to stitch the nucleotides. We know the DNA structure, the double helical structure, which I had just erased now only because I wanted to make space for some other work. We know the backbone of this structure is having the sugar phosphate backbone. The nucleotides are attached to each other, and these nucleotides, that is these nucleotides which are very short, and these are attached to make a big chain, and this attachments that is going to be initiated by the process, that is there is some enzyme which is DNA polymerase and RNA polymerase that is helping us to stitch those nucleotides so that we can have a perfect backbone of DNA helical structures. One backbone, that is the framework, can be made a perfect one with the help of the stitch and the attachment of the nucleotides. So we need these two enzymes for it. These are enzymes which are initiating DNA polymerase and RNA polymerase. Now you may ask, in this case what is happening is the first step we know that it is denaturation that is taking place. This is a double structure, means a double helical structure. Right now I have drawn in this way. The nucleotides are in this form. Now what happens in step 1? It is in this format. In first step, the denaturation that takes place will make it a single strand. That means this strand and this strand, this 5 prime and 3 prime, again 3 prime and 5 prime. Okay, so this is going to be separated, and this separation is initiated known as denaturation, which is taking place in a temperature which is very high, almost 92 to 98 degrees centigrade. You can take around 94 degrees centigrade for 1 minute in a thermocycler. We have a machine for this, so that we can, this denaturation that is taking place, that is separation of the two strands into single strands. Okay, and it is taking place in a temperature around 94 degrees centigrade, and the time duration is only 1 minute. That means the temperature is going to be very high in the thermocycler. Thermocycler is the name of the machine, which is going to have this process done of PCR. It is a thermocycler. It is a machine which is having some wells made, and into these wells this tube, which is, you can say, the PCR tube, which has got a lid type of thing. It is poured in all the material. Now what are the materials that are put in? The Taq DNA is going to be there, the primers are there, the DNTPs are there, and our target DNA, a buffer solution is also there, some oil is put. So these are all going to be filled into these wells, and then the temperature, it's going to run with the help of electricity. Now this point I will come to it in my next video, how the mechanism is going to take place. Let us first understand the steps. Now the requirements, as I have said, we need DNA polymerase to do this process, and the particular temperature that is to be initiated in the denaturation process is, it is for 1 minute 94 degrees centigrade temperature when the strands are separated. Next is the primers. The primers are going to add in the second step. So in the second step, what is happening? Primers which are having, you can say the primers are short nucleotide sequences. Primer. These are short nucleotide sequences which are going to be added. Now where it will be added? The question is where it will be added. If this is 5 prime 3 prime, usually short nucleotide primers, primer adding, that is known as annealing process. The second step, that is annealing, will take place in this matter. Like one is going to add over here, and then further this will be extended, and one is going to be added over here. So primer annealing will take place. This is the second step. First step was denaturation, we know this, and in the second step, when the primers got added into it, these are short nucleotide sequences, and these short nucleotide sequences are having complementation. And the second step, that is primer annealing. Primers, what are the primers? Short nucleotide sequences which are having complementary base pairing rule. Now when I say these primers, let me draw it in a proper manner so that you get it very well. I said that these primers must follow the complementary base pairing rule. Now complementary base pairing rule means I have to know this target sequence. If my target sequence is unknown to me, then I cannot make the complementary base pairing rule go with it. So to have proper primer annealing process to be done, I must know the complementary base pairing rule. So in target DNA, PCR reaction, polymerase chain reaction can never be successful unless and until our target DNA sequence is known to us. This is a very important point that we need to understand. Our target DNA, whichever DNA we need to amplify, we need to make multiple copies, the sequence must be known to us so that the primers can be added. Now primer annealing is a very important step. Now the temperature initially it was raised to 94 degrees centigrade in the thermocycler. That means this temperature is a very high temperature, and the DNA strand will be separated, denatured, single strand now we have got, and the primers, when it anneals, the temperature that has to be maintained in the thermocycler is not at 94 degrees centigrade. It has to be lower. The melting temperature and the primer annealing step, that is primers, when primers are annealed, primers are short nucleotide sequences. These are short nucleotide sequences which are complementary to the target DNA. When these anneal means these are attaching to this single stranded DNA strand. The temperature that has to be maintained should be around 55 degrees centigrade. You can take 50 nucleotide sequences are going to be attached following the complementary base pairing rule. The first requirement is the temperature. The temperature should not be high. Again, the temperature has to come down, because in the first case the temperature was 94. Let me write here so that it is very assured that in the first case it was 94 degrees centigrade. In the second case again it was for 1 minute. That means denaturation temperature is 1 minute, 94 degrees centigrade. I am rubbing this place so that it is not creating any further confusion. And in the primer annealing process, the temperature range goes down to 50 to 55 degrees centigrade when the primers, that is the short nucleotide, oligonucleotide sequences are attached, and the time duration is that time 45 seconds. So you can see the difference got separated, melted, you can say. We had single strands and then the primers annealed. The temperature now came down to, it has to be brought down to 50 to 55 degrees centigrade. The time duration, initially it was 1 minute when the temperature was raised for denaturation. When it came down, it has to be brought down to 45 seconds duration. So you see that the transition or the change has to be done. But once you load everything, you cannot check. Now this point, is target DNA sequence should be known. This is a very important point. Without this, the PCR reaction is not successful. Now after that primers are annealed, what is the next step? The next step is these primers which are carrying the short nucleotide sequences, these are going to be extended, or extension process will be taking place. So after this, the primers, once these anneal, it is the role of tag DNA. This is our single stranded DNA I have shown here, and following the complementary base pairing rule, I have my short nucleotide sequences which are attached. One important point that I have missed to say over here, there are two ways of attachment. One is forward, another is reverse. This is going forward, so it's a forward, and this is in the reverse manner it is going. So what is happening in this case, the primer annealing, this I will cover in my next video, the details about this forward and reverse, what is exactly happening. I will go into it in my next video. Right now this is this point, and it has to be attached in the 3' hydroxyl end. This primers, when it anneals, another point I have missed out, that is it happening in 3' hydroxyl end only. Why in this end? Why it should not take place in 5'? There is a space. Just a short hint I will give you. Say that the DNA strand is a very long strand, and the nucleotide sequence, that is the primer designing, I have a short strand. Now what may happen is that there can be 4 or 5 places when the primers are attaching to their own cells. That means primers are attaching, one primer is getting attached to another primer only. One short nucleotide sequence is attaching itself to another short nucleotide sequence, and it becomes, primers become dimers. So the purpose is not served. This may happen. That's why the primer annealing takes place in the 3' hydroxyl end. It can free only the 3' hydroxyl end. This is the first point. And the second point is that when it has to maintain the complementary base pairing rule, making sure that the GC content is not there too much, because we have to avoid the GC content. We have to place more of the GC in the 5 prime to worry about. We have only 3 steps. The first step I have discussed with you is denaturation. The second step was primer annealing, when we had to remember that it has to be in the 3 prime hydroxyl end only. It can free only in the 3 prime hydroxyl end. The primers which are short nucleotide sequences getting attached to that end, and in the attachment has to be taken place in the forward and the reverse direction. This has to be kept in mind. The GC bond should not be kept in the 3 prime hydroxyl end. The reason is just a short hint, because in my next video I will be discussing this, that if the primers are attaching to their own cells, that is a primer is attached to a primer only, then it is making a self complementary stand, and we do not want that to happen. Next is the tag DNA role. Now what is this tag DNA? The role of this tag DNA is that when the primers, these are the primers which are attached, and these are following the complementary base pairing rule. Now, after attachment of these primers, it has to be extended, and that extension, it can happen with the help of these tag DNA, which is going to extend this line, you can say. These sequences are getting stitched to bacteria which is found in hot springs. Tag DNA Thermus aquaticus is the name of a bacteria that is heat resistant. It is able to withstand high heat, because we know the temperature in the thermocycler is going to be very high, and at that temperature that polymerase that is very active is this Thermus aquaticus. This Thermus aquaticus, you can say, is helping us to withstand this temperature, high temperature, and it is adding one by one all the DNTPs. What are the DNTPs now? DNTPs, let me rub this portion so that I can write what are the DNA polymerase enzyme, that is means a bacteria which is living in the hot spring, and it is isolated from them, that is this enzyme tag DNA Thermus aquaticus, you can say, and it is going to have, it has the ability to withstand high temperature. So what is the role of this tag DNA? The tag DNA, that is this very name, tag DNA or DNA polymerase only, the name of the enzyme, isolated from the bacterium Thermus aquaticus, which is able to live and withstand high heat and temperature. So this enzyme is making all the nucleotides being stitched together, all the nucleotides, all the DNTPs, you can say, all the DNTPs. What are these DNTPs? These are building blocks of DNA. The DNA framework, the building blocks, means it is all made up of the DNTPs. What are the DNTPs? 4 type of DNTPs: DATP, DGTP, DCTP and DTTP. So it is DATP means DNTP, first of all, deoxynucleotide triphosphates, DATP, that is D for deoxy and adenine triphosphate, guanine triphosphate, cytosine triphosphate and thymine triphosphate. These DNTPs are all stitched together and making the building blocks of the DNA, and all are going to be attached with the help of this tag DNA together, and this strand in the last phase, it is extended, that is the process known as extension takes place. So in the first process, first step it was denaturation, second step primer annealing, and in the third step what happened? These DNTPs are linking or attaching all the DNTPs and making this strand long one. So it is, this step is known as extension or elongation, also you can say extension, and the temperature range has to be 72 degrees centigrade. So initially it was 94, then it came down to 55, around 55, 94, 55 and again 70 or 72 degrees centigrade temperature, and the time duration is 2 minutes. So basically PCR is not a worry. There are rest of the matters which has to be very much, and we have covered the steps. First is denaturation, temperature 94 degrees centigrade, melting of the DNA takes place for 1 minute, and then the second step was primer annealing, when the temperature was brought down to 50 to 55 degrees centigrade for 45 seconds, and in the third step when the tag DNA extended the primers annealed over here. See 5 prime 3 prime, this is 5 prime and this is the 3 prime, and we know that the primer annealing has to take place in the 3 prime hydroxyl end only. Keeping in mind there are more number of GCs or GCs should not be kept in 3 prime region. The reason is if they are self complementary, then they will make dimers of their own selves. So self complementary strand should not be made. And then the primer is annealed, making complementary base pairing room following it, and then the DNTPs extended this strand. Now what is happening is basically this is the pure strand. This is also a pure strand, but this one is a hybrid one. So let me make a fresh drawing for you so that you can understand how exactly it is happening for us to understand it better. A diagrammatic view, you can say. I have done this. This is only for the basic for better understanding. See here, this is the DNA strand, original strand, a double helical structure which is helical. Right now I have drawn it in a very straight manner. These are the primers. So this is the DNA segment or strand, and these are the primers which are having the short nucleotide sequences to oligonucleotides, primers or nucleotide sequences it is carrying. And this is our tag DNA, what we were talking about. The tag DNA has the capacity to withstand high heat, and when we are loading it in the thermocycler, it is not only these basic things, there are DNTPs also. There is a buffer solution also, there is some mineral oil also. After this, after 1 minute, that is in the thermocycler, we put all it is a reaction mixture over there. We put all our components there in the thermocycler, and then we run it at the temperature of 94 degrees centigrade. When these two strands, these DNA strands, 5 prime, 3 prime, 3 prime, 5 prime, separate. And these are separated. Now what happens this is the first step, that is denaturation takes place. Temperature 94 degrees. We found that the two strands got separated. The primers, which are short nucleotide sequence, are getting attached at the 3 prime end. The reason also we know. This is our second step, that is primer annealing. In this case, the temperature has been brought down to 50 to 55 degrees centigrade and the time duration is 45 seconds. And from the second step we move to the third step, that is we find the DNA strands. Right now the primers have annealed in forward and reverse manner. The primers have annealed, and tag DNA attaches one by one all the DNTPs, DATP, DGTP, DCTP and DTTP. All these DNTPs are attached and making a long strand, and this is a hybrid one. So this is an extended strand, and the process we know is that is elongation. You can also say extension process, and the temperature that has to be maintained in the thermocycler for extension process, the temperature is 72, around 72 degrees centigrade. Time duration 2 minutes. So we have now at least from 1 we have 2. So this is in the first cycle. Now what happens is, in the first cycle, this is the result, that we have one pure strand, this is a pure strand, and this is a hybrid strand. So altogether we have 2 pure strands and 2 hybrid strands. In this way, after many cycles, after 2 cycles, the scene again will be from here, again you will have like this 2 more, you will have again 2 more you will have. So what really happens is, the strange part of the story is, there will be a time when there will be many cycles. After many cycles, what really happens, each time after each cycle, you are getting two pure strands and two hybrid strands, and that pure strand becomes the template strand for the next cycle. So usually what is happening is, the main strand or the pure strand is our target DNA, and this target DNA and primers, when it anneals, that is this annealing process, what is happening each time, it is happening just after the target DNA. So after the target region, we are having some extra region where the primers are overhanging over there. So after many cycles, the result is that you are having pure, instead of having pure, you are having whole lot of hybrid only. Only after many cycles, the result is this, that you get a pure DNA strand, because each time one cycle of one cycle of PCR is getting completed, you are getting two pure and two hybrid, and this hybrid again will be produced in the second cycle, in the consequent cycle. So main theme, what is happening is, the target DNA is coming in the middle and the attachment is taking place after it. So after many cycles in the thermocycler, the result is in our favor, that we have a pure target DNA is a pure form. Before that, it all becomes, attachment of the primers becomes, after many cycles, the target DNA we are getting in pure form, because after each and every cycle we got our target DNA in the midpoint, and overhanging region was the primers which are getting annealed, that is attachment. So after many cycles, after 30 to 40 cycles, when you have really multiple number of copies of our DNA, then you are able to get your pure target DNA, and that also in a good manner. Now this is one point. Another graphical representation, if I draw, then how do I draw? The first step is the denaturation process, when I am putting it like this in 10 degrees, 20, 30, 40, 50, 60, 70, 80, 90, 100. So 94 will come somewhere here. Now this temperature duration, I have tried to make it a simplified version, because I have to manage with 1 minute and 45 seconds. For 1 minute, let me put another color so that it is clearly understood. Here I am putting degrees centigrade, this is the temperature range, and here the time duration. Now what really happens is, first we know denaturation, it has to be somewhere here. That is denaturation time, the temperature range is 94, around 94. Again, so the graph will go very high. At this moment, it has to go very high. And this is the denaturation, step 1. Again, it has to come down to 55 degrees centigrade. So somewhere here, it will come down for annealing step, the second step, denaturation, annealing, and the third step we know it has to be around 72 degrees centigrade. So you can take it like this, and that is for 2 minutes. So if the third step, that is extension. So finally the third steps are nothing, the 3 basic steps are very clear, that is denaturation, annealing and extension. When the DNTPs are attached, all the DNTPs will be attached and making the backbone or the building block of the DNA to extend that strand, and that strand that we get is a hybrid strand. And each time we are completing one cycle, we are able to get 2 DNA strands which are very pure and other 2 hybrid, and after many cycles we are able to get our target DNA, because each time the target DNA is found in the middle point, and at the either ends we found our primers are getting attached. So there are some other factors also, like primer, designing of the primer how to design, and how we have to create the atmosphere in the thermocycler, some of the detailed study about this PCR, because PCR steps are nothing but the other requirements, other points that have to be kept in mind, that I will do in my second video. So I hope just you have got this idea. PCR chain reaction is absolutely a crystal clear reaction. Keep supporting me so that I can come with more videos on biotech and genetics topics, and further coming up will be zoology and botany topics also will be designed. So keep supporting me, be with me, and if you are not attached to my channel, please visit my channel. Enjoy some time studying with me. Thank you.